Hello and welcome everyone to Bio-Rad's flow cytometry series presenting a webinar on advanced topics in cell sorting. My name is Melissa Ma and I'm the Global Product Manager for the S3 cell sorter at Bio-Rad and your host for today's webinar. First, let's discuss a little bit of the webinar logistics. So the webinar will last approximately 1 hour. Dan Fox from Propel Labs will be presenting on the general concepts in cell sorting, sort modes, cell sorting performance, and discuss sorting speeds. Following the presentation, we'll have about 15 minutes of live questions and answers with Dan. Throughout the webinar, please definitely feel free to use the Q&A box on the left side of your screen in order to submit any questions relating to cell sorting to us. We will get to it and answer as many questions as we have time for. However, if we cannot get to your questions today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, just a little bit about our speaker, Dan Fox. Dan has over a decade of experience in designing high-speed analyzers and cell sorters. During his career at Cytomation, Dako, Beckman Coulter, and currently at Propel Labs, he has led the architecture and design of 3 generations of cytometry hardware. In addition to leading the R&D team that developed Bio-Rad's S3 cell sorter, Dan also designed the electronics and firmware at the heart of the S3. The title of Dan's talk this morning is Advanced Topics in Cell Sorting. I know everyone's excited to hear about it, so let's get started. Dan, you're up.
Thank you, Melissa. I'm really excited to be giving this today. I love talking about cell sorting, so I'll pretty much jump at any opportunity. We've talked a little bit about what we're covering. What we're really doing is we're coming in past the point of analyzing the cells and going over technically what is happening with the whole sorting process. And that's all starting with the pieces that make it happen. So we have the process that needs to happen to get droplets into the sort receptacles. To get up to that point, the first thing that needs to happen is we need to break it up into droplets. We have a solid stream that's being jetted out of the nozzle. That has to be broken up. And we need to be able to individually pick off those droplets and put them where we want them. That's the charging process. And both of those come from the nozzle. As that charged droplet goes down, it enters the electrical field so it can deflect. We're doing electrostatic deflection, so the deflection plates are creating the field for that. And all the rest of the pieces that have to put together the timing, put together the signals, are done with the sort electronics. To jump in and look a little bit at the nozzle assembly from the S3, nice, good view of it. It's always fun to look at it on the computer when you see everything in real life. It's so tiny and hard to really get a good look at. The crystal is what's creating the droplets. And as it's shown here, the top surface and the bottom surface would have a metal layer. And between that would be the piezoelectric material. So as the voltage is applied across that piezo material, it would be expanding and contracting vertically. And that's what forces the wave on. The sample line carries the sample in and takes it down and injects it where it can be hydrodynamically focused to be pushed out of the nozzle tip. And the charging pin passes through the body of the nozzle to contact the sheath solution so that it can put a charge onto the charge carriers, which is basically the salt in the sheath, so that we can charge those droplets. The process that's going on for that starts with the interrogation point up at number 1. And that's where the cells are coming through. They're getting hit by the lasers, they're scattering light, they're fluorescing at different wavelengths, and all the analysis happens. From that point, the cell is moving at high speed, about 20 meters per second, and it's progressing downwards to where it's shown as the break-off point. At the break-off point, the acoustic wave that's being pushed down onto the standing column from the piezoelectric crystal is forcing those droplets to break off. As those droplets break off, we want them to break off with a charge. So before they break off, we use the charging wire to apply a voltage. If we want a positive voltage, we obviously put positive on it, and we can do negative, and that gives us 2 different charges for 2 different directions. By leaving that constant, the droplet breaks off with the charge, progresses down between the deflection plates, and they have a static DC electric field. And that field repels like charges, attracts opposites, so some of those particles, some of the droplets that are charged will go one way, the ones with the opposite charge will go the other way, and as that gets down to where the waste receptacle is, we can leave them uncharged, have them go to waste, or have them charged and go into either receptacle. And that lets us put any combination of droplets in either the left sort, the right sort, or let them pass through into waste. To jump in on the droplet drive portion of that, we talked about there's a piezoelectric crystal that's vibrating, and that's forcing standing waves down onto the column of liquid. Those standing waves couple the energy through, and because it's coupled and forcing it on there, the droplets are going to break off. And by driving that energy, we can make them break off at a very stable and predictable point. Now, the timing of where that point is, we get into later. But the important thing is that by driving this signal, we can make them break off just where we want. If you just spray liquid out of an orifice like a garden hose, it'll break off, and you'll eventually end up with a bunch of uneven large droplets. But by doing it very consistently and forcing it, we can make it break off exactly how we want. That sine wave, which is approximated here, maybe not a perfect sine, but is what creates everything. And we can talk about the different properties of it. Where we're talking about the period, that's 1 over the frequency, and that's really the time per droplet. And so as we drive that signal, every time we're driving one signal onto the crystal, we're eventually going to have that turn into 1 droplet. The amplitude varies by nozzle, varies by temperature, but that is really what is driven and what is maintained, and later we talk a lot more about that. But in the end, the most important thing is the frequency. As it turns out on the S3 cell sorter, with the pressure, which gives us the velocity, and with the orifice size we end up running between 37 and 50 kilohertz. And the 37 to 43 kilohertz is really the typical range that we get into. And all of that comes back from some of these initial equations to what we're looking at. And the orifice that we have on the S3 is 100 microns. What we end up with driving that at 30 psi, which is chosen mostly because it's a very good point when you have a lot of the cells you want to run for typical use. 100 micron orifice, 30 psi is gentle enough for the cells that you get good viability. It gives you, in this case, mathematically it comes out to a nominal 44 kilohertz, which is enough to give you a good high-speed sort, good viability, and it works well with all of the pieces that are there. And the 100 micron orifice is a little more resistant to plugging than can smaller ones be. So that really gives us a good overall range, and we end up with at 30 psi and 100 micron orifice at about 20 meters per second. Now, as it's breaking off, this is always one of my favorite things to look at. The image that you see here is created by strobing a light in front of the break-off and observing it with a camera from behind. So what you're really looking at is the shadow of a strobed image. And when you see it on the instrument, it always looks like it's just up there displayed, but it's really a live image formed from all of these strobes. So once per waveform of the drop drive period, we fire the strobe. And it's fired for a very brief period of time so that we get a nice crisp image. And by firing that at the same point every time, we're looking at one little portion of the period of each drop drive. And so that gives us a fixed image relative to the drop drive so we can see the different things that are happening with that. So what we're looking at doing is setting up the cytometer so that we have the last attached droplet where we want it within the break-off region that the instrument is all pointing at. And then we can measure the properties of the satellite, look at where the detached droplets are, and later in the process maintain it so that it stays exactly where it is. I always like to look at this because it gives you a really good view into how those droplets are breaking off into what's happening there. To get into the droplets and to make sure that they're charged the way we want them, the charging wire that connects up into the nozzle body allows us to put the charge on the entire stream. So when we're charging, the entire stream picks up the charge from the voltage that's being driven onto the charging wire. And that is held constant while the droplet breaks off. And once it breaks off, it's isolated. There's no way for it to go anywhere. There's no conduction path. So it'll hold that charge as it goes down through between the static plates. And as we've talked about, we can do any combination from any droplet. So in between, while the droplet is preparing to break off, in between the break-offs, that is, we can change the potential on that charging wire, and that'll give us a different potential down on the droplets. So by setting the charge through the voltage, holding it constant during the break-off, we always know what that droplet is going to break off with. And that allows us to have nice, stable voltages to give us nice, stable charges on those droplets so we can make sure we hit the sort receptacles appropriately. The most important thing that you end up with in cell sorting is the drop delay. And what that is the time from the laser interrogation to where it's actually going to end up down at the break-off point. And it is the most critical factor in cell sorting because you're going from the analysis world of hitting it with the lasers and collecting the data into needing to put that particle into the droplet that you want. If that time is wrong, we cover this a little bit more later, if that time is wrong, you will be making incorrect predictions and you will not be ending up with what you want. And there's a lot of work over the years that has gone into measuring that, everything from microscope slides to some of the new automated systems like the S3 has that directly measure it, and other systems that estimate it. But that time is a tremendously critical factor in cell sorting because for every percentage you're off, you start to lose that much in your cells. And everything is based in terms of the original drop drive. That is what is setting the overall timing for the whole system. By driving that piezo crystal in fixed periods, the strobe is based off that, all of the timing is based off of that. And by basing it off of that, it ends up a little bit more resistant to anything that you would get in velocity fluctuations because everything still ends up directly tied back to that drop drive frequency. And as we talked about it being the most important thing, this is some great data from Karen Helm where she intentionally incorrectly set the drop delay and then looked at what happens through the process of doing that. And the first thing that you see is your recovery starts to fall off. And that's where I was talking about. If you are off a certain percentage of your drop delay, you will start to immediately be making incorrect predictions on the edges of what you thought you were getting, and you'll be losing some of those. Now, the purity in this example is holding higher until it goes off and then falls off rapidly. And what's going on there is that at lower event rates, even if you're losing some of the cells and getting decreased recovery, you're still not affecting your purity quite yet. A good way to think about that is if you're doing 1,000 cells per second, but a 40 kilohertz or 40,000 droplets per second, on average there's 40 drops per particle coming through. So even if you missed it and it was in, you sorted drop 1019 and it really ended up in drop 1020, there was still no other contaminants that ended up in the drop you did sort. So what you'll see is your recovery was lower, and we define all of these things later. Your recovery is lower because you didn't get the particle you wanted, but your purity is still hanging out up there okay for a while because you're not getting as many contaminants in. It really comes down to probabilities at that point. And if you had a 1 in 40 chance of actually getting the contaminant, you're not nearly as likely to see the effect early on in purity at slower event rates. If you were to raise the event rate way up and do 20,000 particles per second, then your purity will track much closer down to your recovery. There's one more thing that makes the whole deal with all of this a little more complicated, and that's the charge phase. That from the instrument, there isn't really a defined, it's not an integer number of cycles from where the cells pass through the laser until the droplet is actually breaking off. So we have to time that offset between the drop drive that's being driven and where the charge pulse needs to go so that we accurately center, and that gives us the stable period of time for the voltage during which that droplet's breaking off. And the charge phase is just shifting the charge pulse relative to the drop drive waveform, and we can look at both edges and make sure we're nice and centered. And once the charge phase is nice and centered, you know you're at a nice stable voltage so that every time you're going to break off with a consistent voltage and thus a consistent charge on the droplets. And that's what's going to give you really good performance later when you're getting your deflection, to have it nice and consistent. The other important thing about all of this is the maintain mode. It's once you have these parameters on the stream set up and you have the charge phase in the correct place, you have it breaking off where you want it, you're happy with the frequency and the amplitude, you need to be able to keep the stream right where you need it. And we call that maintain mode. And what happens is over time, which can affect all the different properties inside the nozzle body, and most importantly, temperature. The temperature effect on the liquid is very large, and the viscosity changes will tend to drive your breakoff point up and down and affect it all over the place. And so the maintain mode monitors from that strobed image, feeds back parameters, and it can control things like the system pressure. It can control the amplitude that's being driven to the nozzle, so that as the viscosity changes, we can change the amount of energy being driven to the nozzle. So we'll go ahead and jump into the first of the poll questions here. And these are just kind of fun ways to make sure we see what you guys are thinking and how it's going for you out there. So go ahead and check whatever applies here for your biggest challenges with cell sorting. We'll give everybody just a few minutes, and then we'll see how people are feeling about things. So go ahead, lock in your answers here. Looks like people are getting it done, and give just another couple more seconds, and we'll proceed. Find out. All right, so it looks like here we have kind of a tie between not enough trained operators and it being a time-consuming process. So that's great. And it's nice to have these feedbacks so we kind of, as we go through the presentation, we get a feel for where everybody's at. So far to this point, we've just talked about sorting the droplet we want. There's a lot that goes into how do you decide what droplet it is that you want. And that's where things start to get a little more complicated as you start to get into all of the different sort modes. And the question always comes up, well, why do I need these sort modes? What it's really trying to do is you're trying to tell the instrument what's important. Am I really concerned with contamination, or am I really concerned with getting every single cell possible that I have? If you want something that you're going to grow up, you might be very concerned with purity. And if you were doing something very rare like circulating tumor cell research and you have 1 in 100 million or 1 in a billion, well, you certainly don't want to miss it when it comes through if there was a contaminant. And so this is why we have these sort modes, and we try to set the instrument up so that it knows what it's trying to achieve. And as we go through them, the simplest one to think about is enrich, and just says, okay, give me everything of this type of cell. If it's in a drop, I want that drop. I don't care what my purity is. I want every particle that comes through to end up in this tube. Purity is where it starts to get a little more complicated. It says, I want these particles, but don't compromise the purity of my sort. Single mode is great when you need exact counting. I want 1 particle in this well so I can clone it, or I want exactly 100 in here for whatever experiment I'm going to run. And it gives you much tighter control over the counting statistics. The other thing the instrument has to do is figure out what to do when it can't give you what it is that you're asking for. There used to be, aborts has been a good term. It's kind of merged into 2 different things over time. Originally, instruments were doing analog processing. They had a period with which they had to charge up capacitors to the peak signal or charge up integrators to look at the area signal. And you wanted to make sure that between cells or between lasers, if they were spatially separated and had a delay between them, that you had enough time to look at for the entire peak, or you had enough time to integrate for the whole area so you didn't compromise the CVs and the performance of your data. And by having an open window, you had the issue where you could have coincidences. They were called coincidences, hard coincidences, electronic aborts, hard aborts, a whole range of terms for them. Mostly it meant that 2 particles came through and they couldn't be looked at individually. Now, newer systems like the S3, that can be reduced or eliminated, and on the S3, there just simply aren't any coincidence aborts. We're able to look at everything that comes through that is above the threshold as individual events, process them, and have the benefit of storing all of the data before and after digitally so that we can look ahead in time and back in time to get that full area processing. So the electronic hard aborts have disappeared now on these newer systems like the S3. The soft aborts still exist. What those are is where you told this system, I want this population of events, but because of your logic, it can't make it happen. You're doing purity, and there's a contaminant in the drop. And all of that leads down to the efficiency, which is really saying how much of the population that I wanted am I getting? So if all of these were the cells I wanted, what percentage of them am I getting with my current sort setup? And efficiency is a great way to really see kind of how the instrument is feeding back to you how your application is running. Now, for looking at when do we want to take them in the different sort modes, it again gets one more layer complicated. You have to start predicting where within a droplet is the particle going to fall, and what do the adjacent droplets look like. For the S3, all of that prediction logic is done with high resolution and is looking 1 1/4 drops on both sides. And as I've mentioned before, the drop delay is critical. The reason why, as you're predicting and trying to discover and calculate where these particles are going to end up, if any inaccuracy that you have in the drop delay immediately goes directly into that prediction. If you're off 1/4 of a drop, you're going to think it's going to be 1/4 of a drop off. It's just a direct transfer of that number. So by being able to start with drop delay in the right place, that's really the only way to end up with a good sort. And that's a huge thing, to have an accurate drop delay measurement that's really empirically measuring what's happening in the instrument so that you know you have it, and then going forward into maintain mode to make sure that you keep it where it needed to be. The process that the instrument goes through for every particle that comes through is it calculates which of all of the sort modes that are possible does this target cell meet. Then, as all of the cells have come through, it starts to look at the next layer. Okay, given all of these cells, how does that fit into the droplet world now, which can have multiple cells per droplet? Now, do these droplets meet the sort mode that has been selected for the particle that was within that droplet? And then it goes the next step, which we have that we've been calling extend mode. And that says, can we maximize recovery by making sure we don't miss the particle as it's coming through? We have a little more on that here in just a second. Jumping back in to get more details on these sort modes, single mode is pretty easy to explain. You want exactly one particle. To sort the droplet, it has to have one particle in the center half of the cell, and that gives you the best knowledge that it's going to end up where you want it. And to make sure there's no contaminants, because you only want that cell, you can't have any contaminants in the outer quarters of that drop or in the adjacent quarters. Because we've talked about, we're predicting where these particles end up. Prediction is great, but let's say a particle ended up exactly mathematically between drop n and n+1. Well, really, there's a 50% chance it's going to end up in n and a 50% chance it's going to end up in n+1. So when we get really concerned about the purity, anything that's an adjacent contaminant, you worry about it migrating into a different drop. So for single mode, we only sort under very rigorous conditions. And that's where there's the exact one in the center and there's no contaminants possible in that drop or in adjacent drops that could migrate in. Purify mode has a lot more going on. We only want to sort the droplet when the particle of interest is within the drop we're looking at and there's no contaminants on either side. And that's a simple enough case. Fairly similar to single, but it can be anywhere within the droplet. But for Purify, we have the extend mode that's using dynamic logic where we're trying to say, let's be greedy. We want to give the customer everything that they can get into that cell. If they said they wanted it, it needs to go in there. That's the whole point of the sorter. So if that particle is in the outer quarter, we look and say, okay, if it's in the outer quarter, there's some chance that's going to migrate into a different drop. So can we take 2 drops? If the follow-on drop is empty and the next drop has an empty adjacent quarter, so we know nothing is going to migrate into the other one, we can take 2 drops. And we do it dynamically. It's not a requirement. We will sort 1 or we will sort 2. We don't require it to be both, as both don't have to be sorted. So sometimes you get 1, sometimes you get 2. And what that does is it maximizes recovery without jeopardizing your yield and giving you more soft aborts. And the same thing happens in both directions, above and before. Finally, there's the enrich mode. It's similar to Purify, except it's less restrictive. You can have any particle within the cell of interest as long as there's 1 positive. Similar for the follow-on, it's about the same as Purify, except we don't need to check the adjacent quarters because we aren't as interested in those contaminants migrating in. Really, in enrich, we're trying to just make sure we give you all of the cells possible. So we extend where we can if it's in the outer quarters, but we aren't as worried about the contaminants. In the extend mode, talking about the dynamic logic behind that, that's pretty unique. Some of the other systems, you can set it to sort 2 or you can set it to sort 1, but by being able to let the instrument make the decision about what is best for your sort, it really helps keep that flexibility. It makes sure that you get the cells that you want without jeopardizing your yield and the efficiency as reported by the instrument. The mixed mode sorting is another handy feature. is that there's times where you have a really precious sample and you want a high purity output, but you don't want to waste any of the cells that are coming through. So mixed mode allows you to do purify left and enrich right, or the other way. But really you can start with one region and say, I want my high purity stream one direction. Anything that would abort, put it in the other tube. And because you can set it up that way, you never waste any of your sample. And this jumps us into the second poll question here about what applications you're typically running. It's just another, like I said, a good way to see who's on the call, kind of what things we're looking at and where we can point some of the questions later towards. So please go ahead, take whatever seems relevant to you, put your answer in, and we'll give just a few more minutes here for some of the people that are still getting their answers set. And then we'll jump forward and take a look. So here we go. All right, looks like a lot of cancer and tumor biology going through, also transfected cells, and a pretty good spread here across the way. Now, we've talked a little bit about purity not being a great metric of performance. And there's a lot of things that go into that. The number one, which actually is performance, we've talked about, is drop delay. Having that set will directly go into purity, but only at high speeds. There's a lot of other things that do directly go into purity regardless of the instrument, mostly related to the sample. The preparation and the quality can have a huge input. You know, if you don't have a good sample going in, you're not going to get a good sort coming back out. And the rest of that comes down to the setup, positioning, and how fast you're going. The faster you go, you can start to have more doublets that come in, and depending on what it is you're sorting, you may affect your purity through that. But a lot of people jump right to purity, and I always try to get people to back up a little bit from that, because when you're seeing purity, a lot of the times you need to look at the sample before you go to the instrument. Or you can verify the drop delay and then go back to the sample, but usually it's sample related. And we've been using some of these terms, and so we wanted to get them in and define them well. And all of these terms, recovery, yield, efficiency, have their own little nuances depending on who's talking about them and where they're being talked out and which company it is. And it makes it a little tricky out in the field. But with recovery, what we're talking about is how many particles did you get in your sort collection tube compared to the number the instrument told you it sorted. And it's really that simple. If it said it sorted 10,000 cells, how many actually ended up in your tube that you wanted? And yield is the whole thing from beginning to end of how many did you end up with versus how many were originally in the tube that you were putting on for your sample. And that's where things can get pretty tricky, is with yield there are so many different factors that go into it that can really make it a little bit troubling. And so we get through some of the different ones. And here's the definition of recovery, is that we're really just looking, again, like I said, how many did we get versus how many were possible that the instrument saw. And so this is a very easy thing to assess. The instrument told me it sorted 10,000. You can go through and do a count of what's on your tube. And as long as you do all of that accurately, you can get a very good representation of what your recovery is. And the recovery really has a lot to do with the actual sorter itself. Everything that's occurring from interrogation down to collection. Because recovery is based on the instrument telling you, here's what I saw, and then you can count what ended up being in the tube. So really it's a much smaller piece of the pie, but a very important one. And that's where we've talked about this extend mode being so important, is that we want to maximize recovery. Because if you're sorting, you're concerned with the cells, and we want to make sure that you actually get what ends up in there. Now, the yield is much tougher and really talks about the entire ecosystem from what goes into the tube to the very end of what ended up in collection. And that gets very tricky because there's a lot of things going on to it. You have some losses in the tube and in the line, and then you get into the whole thing about the statistics. And we have more coming up on the Poisson distribution and what that's doing. But basically, when you have random things coming through, you're going to have some proportion that are doublets or triplets, and that's where 2 or 3 are stuck together. Sometimes you might have a clump that comes through. And there's times that as they're coming through, you have contaminants in the same droplet as a target cell, and then you would have that soft abort. And all of those things can add up so that you have a yield from what goes in to what comes out. And some of that is related to the instrument. Some of that is related to the performance. But It also involves the sample itself and the tube, and that's where yield is so hard to look at that efficiency is terrific. It's directly reported by the instrument, and what efficiency is breaking it off and trying to look at a smaller portion of the yield. It's looking at what was sorted out of what could have been sorted, and so it's letting you see how much is lost to aborts. If the instrument is giving you 80% efficiency, for every 8 out of 10 that's coming through, it was able to sort by knowing that the droplet matched the criteria. So with purity, as you have different samples at different speeds, it affects the efficiency. And because that's directly reported from the instrument, it's very helpful and very useful to look at that and to be able to directly feed back into what you're doing and what kind of decisions you're making. Whereas the yield, it's a little bit slower. To really check the yield, you have to do a count on your input tube, sort it, be able to get the output, and then do a recount on it. And it can definitely be helpful. You can do a lot with yield to improve your overall sorting efficiency and to improve the overall yield that you're getting. So a lot of work can be done on how you're collecting the cells, what kind of dilution you're using, how they're going in, how you're running them. But by going back to the efficiency and seeing that number directly from the instrument, it helps you work on the yield. And again, these terms are used in a lot of different ways, reported on different instruments, and some of the instruments are configurable, and you can adjust the number that it's reporting to efficiency based on your preference for how you want that number reported. So when you compare one instrument to another, it can really be very tricky to use the numbers that are coming out of the software. And the best way, again, is to really get down into taking a sample, pipetting it into 2 different tubes, running them on instruments, and then using a third instrument to look at the actual sort collection on the output. And when you do that, that's a much better way to assess comparative efficiencies, comparative yields, comparative recoveries, because you've taken some of those, the software fudge factors out. There's a lot of different ways the software has to try to come up with these numbers, and some of them are even configurable, like I said, and that can just make it very, very tricky. A big question that comes up a lot from going through with all of this is, how fast can I sort, or how fast should I sort? And it's always a tough question to answer because it's so application dependent. And the way the instrument works, and with the electronics processing, all of the acquisition and sort logic and everything else can be made in about 600 nanoseconds, which is way faster than physics will allow. I would always love to push out some sort of physics upgrade package and make everybody sort at 1 million droplets per second. But what you really end up with comes down to all of those coincidences, all of the timing, all of the probabilities for particles ending up with contaminants and droplets and not being able to go where you want them to. And that gets into where you can start all the analysis. The Poisson distribution is a wonderful thing to use for looking at this. It's right up the alley of what we're doing. The Poisson distribution models the probabilities that you will get discrete numbers of events during discrete times, given an overall average rate. It's used for all kinds of things, from traffic. If you have an average traffic flow and you want to know the probability of 10,000 cars within a 10-minute period ending up under this bridge, all the way down to grocery stores using it to try to figure out how many customers could end up within a certain window to know how many registers they need to have open. And in sorting, it's perfect. We have discrete time intervals. Those are the droplets. We have an average rate. That's the rate that the cells are coming through. So we can use the Poisson distribution to run it out into model and to see exactly what we expect. And so that lets the math help give you a good idea of where to run. And instead of having to run through all these equations manually, there's a lot of spreadsheets that are out there that help you get a good idea of where to start. And there's a good example that's here. It's using 1% of target cells out of the input sample and saying, at these different rates, what do we see? And this is where the answer gets tricky. If you want to go as fast as you can see the best sort rate in cells per second, and that middle column continues to increase as you go from 2,500 to 15,000 cells per second. However, your sort efficiency is going down. So you're sorting faster. So if you have lots of sample, you can get the sort that you want done faster by going faster, as long as you have some sample to spare. But if your sample is precious and you're not as concerned with time, but with efficiency, you may need to go slower so that even though your sort takes longer, you end up with a higher sort efficiency. To further complicate that is the fact that the Poisson distribution and all of the math built around it is assuming that everything is perfectly random. If it's not perfectly random, you start to be non-Poisson, as everybody tends to say, and you are clumpier, and so you have higher probabilities of having contaminants in with the cells that you want. And this is where the instrument efficiency is great to look at, because it's going to tell you this is what you're really getting. You predicted that you were going to be at 84%. You're really getting 75%. That's not too bad. You might be happy with that. Or you might want to slow down a little more or put more work into your sample into dilution. Because the closer you can get to Poisson, the closer you are to perfectly random, the closer you are to Poisson. I always like that. It's good. It's easy. It's simple to remember that as long as you can have random data coming through, it's the best for the instrument. Things are spread about as far as they possibly can be on average, and that gives you the best chances of getting target events without contaminants when you're wanting to sort. And of course, that's related back to doing a purity sort. If you're doing an enriched sort, it isn't nearly as important, unless you're trying to maximize the purity of your enrichment, in which case you come back to exactly the same problem. You're trying to pick how fast do we need to be running this versus how much sample am I willing to lose. And some good guidelines that we have is that if you're doing small cells that aren't very likely to stick together, you can start out with a higher concentration, and that allows you to go faster. In the end, what you're really trying to do is match what's a reasonable concentration to the instrument. Most cytometers that are out there, you're running in the 10 to 100 microliters per minute range. And if you're pushing that 100 microliter per minute range, that's really getting up there, and you start to have broader CVs. The inner stream for the hydrodynamic focusing starts to broaden, and it starts to be harder to detect all of the particles with the same precision that you get when it's a nice small sample stream. So keeping that in that middle range of even up towards 50 microliters per minute is a good target. And as you can increase that cell concentration, you can start out running a little bit faster. On the inverse, if they're sticky cells, you want to start out more dilute so that your cells are less likely to be next to each other to stick. And you want to start out a little bit slower because you're going to be running more fluid through. And of course, there's starting points. So frequently you can run considerably faster than these numbers. But it's always a good place to get it on there, start it running, and then really see what the feedback is from the instrument. If you're seeing the efficiency you're happy with, then you can leave it there. If you're really wanting to go a little bit faster, you push it. And sometimes you go through and do a few sorts to really nail down an application so that you're really achieving what you want. Because at the end of the day, it's a sorter. You're trying to sort cells. You need so many in the collection tube for your experiment. And that just backs you into how many do I have to put into the beginning, which may or may not be something that you can tweak. And if you can, then you can go faster, go up to better sort rate, a little bit lower efficiency. And if not, and you have a fixed sample and you need as much as possible, then you want to start looking at running a little bit slower, keep that efficiency up, and then you can play with the concentration if you're having any issues with not having totally random data. So all of those pieces are kind of coming together with the technicalities of how the sorting is done, and then the statistics of how they come in, which then is matched by the sort modes. That lets the instrument know what it's supposed to do to make its sort decision, charge the droplet that you want, and have it end up in the test tube you're looking for. And at this point, we'll turn it back to the audience, and we'll see what kind of questions you guys have and what you're interested in, and answer as many as we can. So, Melissa, back to you.
Thanks, Dan. Thanks for a very engaging talk. Everyone, this would be a great time to submit any questions you have regarding the topics covered in Dan's presentation. Again, the QA box is located to the left of your screen. If any of you would like a copy of Dan's PowerPoint presentation or some other additional resources about the technology, they are also available for download via the links to the left of your screen. This webinar has been recorded and will also be available on demand within the next 24 hours. If you'd like any additional information also on the Bio-Rad S3 cell sorter or on general topics of cell sorting, feel free to reach out to your Bio-Rad cell biology specialist as well. So I think there are a few questions that have come up that are still being submitted, it looks like. But here's a question for you, Dan, if you could answer. Can you provide an example of when someone would use mixed mode sorting? And is there something equivalent with other competitive instruments?
That's a great question. For competitive instruments, the MoFlow family definitely has mixed mode sorting. Past that, I don't remember off the top of my head. I know I've seen a spreadsheet somewhere on how you can get close on some instruments and how other ones you have to define the mode and then it's applied globally. So I don't have a great information to tell you on all the exact competitors. When you would use it is really when you get down to having kind of a rare sample, something that is very important. And I don't have a great example off the top of my head for what that might be biologically, but there's plenty of times we run into customers and they say, look, this is a really critical sample. I only have this many cells. That's all I was able to get from this mouse that I was doing my experiments on. I need a pure sample, but I don't want to lose anything, and that's why we have the feature, is it allows you to get that high purity tube while catching everything else in the other tube, and then you might be able to rerun that and to gain more of them, or you might save that in case you want to redo part of the sort. So a lot of it comes down to really just the importance of the sample.
Okay, great. Here's another question. How do you decide coincidence?
So coincidences are decided based off of the sort mode logic. And a coincidence, which is usually referring back to purity mode, being the most common one that's run, has to do for a soft abort. I guess it comes down if you're talking, some people use coincidence meaning the soft aborts, and some people are meaning it for doublets. So I'll answer kind of both briefly. For the soft aborts, it has to do with that window of droplets that we're looking at. So the coincidence being that with the droplet that you wanted to sort, there was either a contaminant within it or within an adjacent quarter. So the droplet failed the sort logic for purity, was unable to be sorted, and so we would call that a soft abort. For doublets, a lot of the doublet discrimination, which could also be a coincident event of two cells coming through in a laser, a lot of the doublet discrimination can come down to using area parameters, pulse width parameters, and trying to get whatever other information you can to distinguish between the one particle and two particles. And that really varies a lot by application and by sample sometimes. So really knowing what it is that's being run, the doublet discrimination can take a little bit to figure out. I hope that helps answer that.
Okay, Dan, maybe this is an easy one. What is the milliamps typically applied to the stream?
The milliamps I have looked at, and I'm not remembering well off the top of my head. The crystal ends up looking about like a 1 nanofarad capacitor. Typically, it's driven on the S3 system in the 5 to 15 volt range. The actual current that's flowing in RMS terms, because it negates itself back out because you're flipping polarity, it's fairly small. It's down in the single digits of milliamps. Without pulling back up my physical model on driving the 1 nanofarad capacitor, though, I don't exactly remember, but I think it's in that single-digit milliamps. But the 5 to 15 volts, that's really the controlling factor. It's the voltage across the piezoelectric material that's giving you the expansion and contraction. And so all of the current is basically parasitic because of the capacitance.
Okay, Dan, here's another question. Can you provide an explanation as to why flow rates should be increased for smaller cells?
Really, the explanation for the smaller cells is that you want to run as fast as you can to get the sort done as quickly as possible. In general, you're never forced to run faster. But with smaller particles, you have more distance between them. The smaller particles I've worked with also tend to be less sticky, and you're able to pull off higher event rates while still getting good efficiencies. And so typically you want to run faster just because you can, and you get done faster, and you free the instrument back up and get the sorted sample faster to the researcher.
Okay, great. This might be a segue question, but you may have answered it. Does the smaller particle size reduce the efficiency?
In general, the smaller particle size increases the efficiency because they're less clumpy. I'm sure that everyone out there has some great example of when small particles can still be really sticky. But most of the ones that I've worked with, the smaller particles, tend to not be as sticky. and they tend to have to sort with a higher efficiency. That, of course, is very dependent on the sample, so your mileage may vary.
Great. Maybe one or two more questions. The trigger signal marks how is the trigger signal related to the position of the particle in the drop?
That's a great question. There's lots of different ways of looking at that. If you do the leading edge, you run into the case where taller signals are triggering at a different time relative to smaller ones, and doublets can shift the timing on you a little bit. On the S3, all of the timing is based off this, effectively the centroid. It's the peak that's looked at. If you do have asymmetric doublets, that can throw it a little bit, but what we're trying to do is look at the middle portion. Actually, I misspoke on that. We do look at the centroid of the time above the threshold. And so that helps center things out so that you're closer to the center of the cell, and that gives you a more consistent timing later off for looking at the droplet. A lot of older systems just use storing the time at the leading edge, and you can actually play around with different particle sizes and shapes and affect your timing a little bit.
Okay, here's a re-explain question, Dan. Can you re-explain the importance and how the charge phase works again?
Yeah, absolutely. And it's kind of funny because the charge phase is almost like this little thing that's thrown in. All of the other pieces kind of go in this nice linear order, and then charge phase is just this random time between 0 and 360 degrees that ties it back in. When you're looking at it, it's so easy to think an integer number of drops. But the actual time from where the particle hits the laser until it is physically breaking off can be in fractions of the droplets. And the drop delay takes that time out. It knows the time when we actually directly measure it with ProDrop that it's 31.25 drops. And we know that. But that is a little bit different than the exact timing on when you want to start charging and stop charging. So I always think of this as you're looking at this droplet and it's pulling down, pinching off, and as soon as it pinches off and breaks away, at that point, whatever charge it has, because of the voltage given to it, it's going to break away with. And so if you do this in a very bad way and you set your charging to be changing, it's basically a square wave. And so if that square wave is changing right when the droplet is breaking off, you'll get a really hazy, fuzzy, messy defanning pattern, or sort streams. Your sort streams will just be a whole giant mess from left to right because the voltage is changing, and so that droplet can break off with a whole different range. So by using the charge phase, we can just shift the time without changing any other parameters in the system. It shifts the timing on when we charge and when we switch that charge to the next droplet. So once per overall period of the drop drive, we're flipping that charge to be ready for the next drop. And we want that to be basically centered on when that break-off occurs. So if we think of it in terms of time, we might want to have 5 microseconds before the break-off that our voltage is nice and steady, and hold it for 5 microseconds after the break-off. That gives us the stable time that even if the droplet boundaries from one drop to the next, or God forbid there's a cell between the droplets, which can change the timing a little bit up or down, as everybody knows, if that's happening, you want it to be as stable as possible. So by nicely kind of bracketing that charge, we give as much chance to break off with a stable charge as possible.
Okay, great. And I guess also another leading question would be, if you're having trouble creating the nice side streams, would you be then adjusting the charge phase or defanning or both?
The first thing I always do if I have side stream problems is to make sure I'm not running DI water. Everybody has some funny story about being certainly convinced that they were running salty sheath water, and somehow they'd swap tanks, mix something up, and DI water with no salt has no charge carriers, and you will start to go fuzzy, and then you will lose the charge. And you can spend several days working on electronics trying to figure out what it was until you get back around to that. So that's always the first thing to check. When you get past that, of going through some of those wizards to make sure that you have a good droplet creation from the beginning so that it looks right and the system is holding it in place with maintained correctly, at that point, the phase certainly can be important. It's one of the harder things to look at with cameras. It does it very well, but that is certainly the charge phase can be a culprit if you are having side streams that are looking askew.
Okay, and does the S3 have the ability to control charge phase through the software, or is this also automated?
It automatically sets it up. There are administrative panels where that can be adjusted if it isn't doing it well. But all of that under the normal setup where it's going up and automating itself through the process of configuring the stream, maintaining the stream, and then configuring everything for charging and deflection, all of that is included in the automated setup to make sure the charge phase is set correctly.
Okay, for our last question, just a little bit more detail about the S3. What is the smallest and largest size of the sorted particles that can be done on the S3?
The smallest size comes down to what you're trying to detect. If you're having to trigger off a forward or side scatter, I believe we have the specification on the S3 at half a micron. I've certainly sorted smaller things on it than that, but the specification is half a micron. On the upper limit, it starts coming down to the ratio of the particle size to the nozzle size. And eventually, if your cell is too large, you disrupt all the fluidic effects that are happening and forming that droplet. And so you imagine a 100-micron cell coming through a 100-micron orifice, that is going to significantly affect the droplet creation. And a safe ratio is 1/5. So if you stay at 20 microns or below, it's usually very easy. If you go to 5 or 6, 7 on that ratio, it's even safer. 5 is easy, and if you start to go below that, it just requires some testing. Some of the larger cells are fairly evenly shaped, and they don't tend to disrupt droplet formation. Some of them, as they start to be large and oblong or large and weirdly shaped, can start to affect the droplet disruption. And sometimes you're okay with that. We've had people sort some pretty crazy-looking things on there, and they say, yeah, I really don't care if I'm losing some of them, which comes down to recovery problem mostly. I don't really care if my stream's fanning a bit. As long as I'm getting some of them in there, that's all I need. I just want to try to get some of these so I can go off with a higher concentration and do whatever it was they were doing with them. And there's times that's totally fine. So again, it comes down to that application. But if you stick under 20 microns, it's usually pretty easy on a 100-micron nozzle.
All right, thanks, Dan. That was a lot of questions. Anything else that's been submitted and that we weren't able to get to, we'll definitely get back to you personally or via the cell biology specialist in your area. So thank you, everybody, for attending this webinar in Bio-Rad's flow cytometry series today. We will be hosting additional webinars in this series, so definitely please join us again. As a reminder, today's webinar will also be available on demand within the next 24 hours. For those of you who, again, have not had your questions answered, we will be following up with you in the next few weeks. For more information on the S3 cell sorter and educational resources, visit www.bio-rad.com/S3webinar. This URL is also displayed on the left side of your screen in the resource box. Thanks again, and have a great rest of the day.